All right, guys, we are going to start chapter 10. We're on page 169 in your binder. I'm assuming this is the right binder that I'm holding here. Hold on, let me make sure it's 2020. Yes, so page 169. We're going to discuss chapter 10. I probably won't do the whole thing, so we'll do chapter 10 little notes today. And I should have already given you some information about the fruit fly lab that has been kind of disrupted a little bit. And then I'll finish chapter 10 next time I see you, and we'll do just a little web activity. So the bulk of this unit, unit 6, is genetics, and genetics, problems and patterns of inheritance. But before we can get into that, we need to talk a little bit about meiosis and how gametes get made. All right, I am going to show you this video tomorrow. It's actually a really cute video. I love it. These are the things you need to know. Difference between asexual and sexual reproduction, meiosis and fertilization, what those are, what homologous chromosomes are. So be looking out for that word when I discuss it. The words diploid and haploid, how those change through the process of meiosis. The differences between mitosis and meiosis. So I am going to bring mitosis back. Okay, so just be ready for that. And then the importance of crossing over, independent assortment, random fertilization to increase genetic variability. All right, so before we get into the process of meiosis, we're going to discuss some vocabulary words. These are great notes because you don't have to write a whole lot. It's just fill in the blanks. So first you need to know what a gene is. A gene is a segment of DNA that codes for the basic units of heredity. And we've already talked about what genes are, what DNA is, what DNA actually codes for, which is proteins. And so we've got a pretty good grasp on that, I think. Offspring are going to acquire genes from their parents by inheriting chromosomes. And this is just a picture of a family, and what we would discuss is the similarities and differences between the parents and the children. But basically, what I'm trying to get across to you here is that genes are passed down from parent to offspring. But it's not always it's not like asexually reproducing organisms where one parent is going to be identical to one of the children. There's a mixture of the two, right? And so we're going to talk about why that happens. So two types of reproduction, asexual and sexual. Asexual reproduction is going to produce clones, genetically identical. That's like a bacteria that undergo binary fission. That's like the planaria. When you cut a planaria in half and two planaria grow, they're clones of each other. Sexual reproduction involves meiosis, producing gametes, sex cells. So gametes, remember, are sperm and egg. And so meiosis is the process of making sperm and egg, in case you were wondering. Asexual reproduction requires only a single parent. Sexual reproduction requires two parents, male and female. And so if you're looking at your notes, I have these side by side here on your notes. One's on top, one's on the bottom. So asexual is on top and sexual is on the bottom. And you're just filling them in like that. All right. Asexual reproduction leads to little variation in the population. The only time there's variation really is only through mutations. We learned a little bit about genetic variation in bacteria through the process of conjugation, transduction, and transformation. But in general, without those things, bacteria are only going to get genetic variation through mutations. Sexual reproduction leads to lots of variation, genetic variation, and lots of diversity among offspring. The benefit of asexual reproduction is that it's fast and energy efficient. Sexual reproduction is slower and energy consumptive. And an example would be budding. So budding is kind of like what the planaria do naturally. They twist a piece of their body part and break it off. Yeast do that. And then another thing that we've already talked about is binary fission in bacteria as a form of asexual reproduction. Sexual reproduction, students are always like, they're like, wait, trees? What? I'm like, yeah, humans do that, but so do trees and so do plants. Now, it's not like the same like activity or the same way, but sexual reproduction just means there is a sperm from one organism and an egg from a different organism, and they meet to form a brand new organism. And yes, trees do that. So how do they do that? Well, the wind can blow pollen over to flowers of another plant. The pollen meets the ovule or the egg and forms a fruit and the seed. Y'all know how that happens. But yes, plants are sexually reproducing. Just kind of an interesting little tidbit I like to throw in is this word called parthenogenesis. I love it. It's so interesting. And parthenogenesis is when an organism can reproduce asexually when a male partner is not available. So this means that it is an organism like a shark or a bee that should be reproducing sexually, but if a male is not available, they can undergo asexual reproduction and make offspring. Now, who can or which organism can do it? It's only females. So female sharks have been shown in captivity to be able to asexually reproduce and form female sharks. Bees, and when we get to ecology, you're going to learn all about bees. You're going to know so much more about bees than you ever thought you would in your life. But if it is not the queen, there are other females. Only the queen is supposed to be sexually reproducing and laying eggs. But sometimes things go wrong in the hive and the other females will take over and they'll start laying eggs. They can only lay eggs for male bees. And so we'll talk about that. But I just thought that parthenogenesis was something kind of interesting to mention, that there are some sexually reproducing organisms that can go back and forth between asexual and sexual reproduction. Another interesting fact, the New Mexico whiptail lizard, males used to be there, but now they've become obsolete because of parthenogenesis. The female lizards just, just decided they didn't need them anymore and thought it would be a lot more efficient if they just started asexually reproducing. So there are only female whiptail lizards now in New Mexico. Super interesting. Page 170. I'm going to go through a few more vocab words. So we're still not talking about the process of meiosis yet. Let's talk about just general vocab. Somatic cells are body cells that are not sperm and egg. I know I've gone over these words before, so I'm going to go through quick. Each pair of homologous chromosomes include one chromosome from each parent. So homologous chromosomes include one chromosome from each parent. Humans have 46 chromosomes, right? One half came from mom, one half came from dad. There are only 23 types of chromosomes. I'm going to show you something a little bit later. It's called a karyotype. It's something that you should have seen in cell bio, but it shows you what homologous chromosomes are pretty good. So homologous chromosome is the chromosome type. In humans, it's either 1 through 22, and then you have the sex chromosomes, one from mom, one from dad. Autosomes are chromosomes that don't determine sex. You have 22 pairs of those as a human. Then you have your sex chromosomes, X and Y. Females are XX, males are XY, and then we have gametes, 22 autosomes and one sex chromosome. Okay, and you're like, whoa, 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 whoa. Okay, so fill in the blank. Then I want to go over this really quick again. I should have put it in a different order, and I say this every year, and then I end up not doing it, and I get lazy. It's okay. Somatic cell and gamete, those two words should go together. Somatic cell and gamete should go together because they're both types of cells. A somatic cell is a body cell that's not a sperm or an egg. A gamete is a cell that is a sperm or an egg, okay? So that's how I should rearrange this slide, but for the sake of time, I'm not going to do that right now. So somatic cell and gamete go together. They're two types of cells. Autosomes and sex chromosomes should go together because they're each a type of chromosome. Remember, a chromosome is condensely packed DNA. You have 23 pairs of chromosomes to make a total of 46. One set from mom, one set from dad. 22 of those pairs are called autosomes. They have genes on them that don't determine sex, male or female characteristics. Then you have one set of chromosomes. You're supposed to have two, but I'm going to have you probably do an activity to kind of show you how that can vary among different people. But you're supposed to just have two sex chromosomes, one from mom, one from dad. If you're XX, you're a female. If you're XY, you're a male. So that's kind of how you would work through these vocab words. So hopefully that made sense. Let me know at the end of the lecture if you're still confused on that. So remember, homologous chromosomes are a chromosome type. It has the one from mom, one from dad. And so what I'm trying to have you see here in this slide is that chromosomes are going to line up on the metaphase plate a little differently for meiosis than they did with mitosis. And what that ensures is that there are 4 resulting daughter cells. This should be familiar from cell bio. So one of the major differences between mitosis and meiosis is in mitosis, you have one cell that splits into 2 identical cells. In meiosis, you have one cell that splits into 4. All right. And so what happens is you have these homologous chromosomes that line up together on the metaphase plate. All right. So in meiosis, the cell divides twice. The result is 4 daughter cells, each with half as many chromosomes as the parent. So we would call that haploid. And why does that make sense? Well, if a sperm and an egg each had complete sets of chromosomes and then they fused together, they would make a human that had too many sets of chromosomes. If that ever happened, which I'm sure it does sometimes, it would result in an immediate miscarriage, most likely. Humans can't survive with any more than 2 complete sets of chromosomes. Plants can, which we'll talk about a little later when we talk about evolution. It's really interesting. But humans, 2 sets of chromosomes, 1 set from mom, 1 set from dad. 1 set comes in the sperm, 1 set comes in the egg. They fuse together to make a diploid cell. All right, let's talk about the first division. We're getting into the process. We're on page 171 now. The first division, during interphase of the cell cycle, chromosomes replicate. So remember, if you can kind of look in your mind's eye, what the cell cycle looks like. It's that big circle. We're reading it like a clock. G1, S, G2, then meiosis. So it's kind of the same thing. G1, the germ, it's a germ cell. A germ cell is a diploid body cell, like body cell that divides to form sperm and egg. It grows, then in the S phase, DNA is replicated, and then in the G2 phase, gets ready for meiosis, and then during the M phase, it's meiosis. All right, so during the interphase, chromosomes replicate during the S phase of interphase. Prophase 1 is this thing called synapsis. This is different than mitosis. Homologous chromosomes are going to pair up into what's called a tetrad that has 4 sister chromatids. So I want you to look right here. Look where my cursor is, because this is really difficult to understand. You have, let's say this is chromosome number 1. So remember, as a human, you have 22 chromosomes and then sex chromosomes, right? Let's say this is chromosome number 1. The blue one here, and I know that this is like stereotype. Well, the blue's supposed to be male and the red's supposed to be female. Sorry if that bothers anybody, but the blue one right here is chromosome number 1 from dad and its copy, okay? The red right here where my cursor is chromosome number 1 from mom and its copy. Together, these are homologous chromosomes, and they consist of 4 tetrads. All right, it's really difficult to understand, so I hope that you didn't just zone out right now because there was nothing to write. So homologous chromosomes, it's a chromosome and its copy, another chromosome and its copy. They're the same type of chromosome. They line up together to form a tetrad. And then they do this thing called synapsis. What they do at the synapse, do you see how they're kind of like intertwining with each other? They do this thing called crossing over. The site of crossing over, which means the location on the chromosome that breaks off and switches places, is called the chiasmata. So if you look right here, do you see how this red one took a piece of blue and this blue one took a piece of red? They synapsed, they crossed over. The site of crossing over where the chromosome broke off and reattached to the other chromosome is called the chiasmata. So be sure to let me know if that confused you. After crossing over occurs, the tetrad, meaning the 4 sister chromatids, the 4 sister chromatids is what they're called. They line up in on the metaphase plate just like before, and then pairs of homologous chromosomes separate. So look at my cursor. These 2 blue ones are going to go to the left. These 2 red ones are going to go to the right. These 2 blue ones are going to go to the left. These 2 red ones are going to go to the right. However, what has happened is they have exchanged genetic material through the process of crossing over. All right, the sister chromatids are still attached by the centromere. All right, and then what you end up with is a haploid set of chromosomes that has chromosomes with their copies, and each chromosome has 2 sister chromatids. So, yeah, you don't need to write anything down for that. All right. Here's a picture of what I just discussed. In prophase 1, you have your chromosomes condense. The chiasmata, they line up, homologous chromosomes line up together. They switch genetic material amongst each other, and they're all like jumbled up and mixed up. And you can see that in this anaphase 1. You see how this isn't just all red. There's a little piece of blue right there. This leads to genetic variation. This is how you can have some characteristics from your dad's grandma and some characteristics from your mom's father, your grandfather, or something like that. That's how all these characteristics get all mixed up together. It's because of this process of crossing over, and that you end up with a combination of genes that were never there before, basically, right? So prophase 1, crossing over occurs. Metaphase, they line up in the middle. Anaphase, separation. Telophase and cytokinesis. Not done yet. That's meiosis 1, not done yet. Meiosis 2 is the second division that's actually going to create the gametes. Because if you look here, you still have a chromosome and its copy still attached to each other. If you look at my cursor right here, you see these 2 blues. Even though that one has a little red piece on it, it doesn't matter. Those still need to separate. These 2 need to separate. These 2 need to separate. So meiosis 2, you don't have any interphase. You don't have any crossing over. What happens is a spindle forms. Now these ones are going to line up, and this is going to look a little bit more like mitosis. Sister chromatids separate, telophase, cytokinesis. You end up with 4 unique haploid cells. They're genetically unique. So we'll make sure we get all that written down if you need to. Yes, there's a couple of fill in the blanks there. All right, and then you have a picture here that I'm about to show you. So pause if you're not done writing things down because I'm about to click. All right, here's the second picture. So here's this picture right here. You ended up with 2 cells. Now you end up with 4, okay? These are the 2 that resulted from the picture before. Now they're each going to go through their round of division. You end up with 4 genetically unique meiosis, or genetically unique gametes through the process of meiosis. Okay, hopefully that made sense. The events that are unique to meiosis and not in mitosis, synapsis and crossing over, happens in meiosis, does not happen in mitosis. Since it does not happen in mitosis, the cells that result from mitosis are identical. Since it does happen in meiosis, the cells that result from meiosis are genetically unique. In metaphase 1 of meiosis, pairs of homologous chromosomes line up. So you have that tetrad. That doesn't happen in mitosis. And then in anaphase 1, homologous chromosomes separate. And so the sister chromatids are still attached. So this is meiosis 1 shown in this picture right here. So remember, these still need to separate, these still need to separate, these still need to separate. So this one's going to split into 2. This one's going to split into 2. And because of crossing over, they're going to be genetically unique. I know I keep repeating things, but I'm just hoping that it helps a little bit. Okay, that is meiosis. Hey, all right. And still, you know, the things that were true about mitosis still are here. There's still kinetochores and centromeres and stuff like that. And it's still the same, you know, metaphase is lining up in the middle. Anaphase is pulling apart. Telophase is the dividing of the nucleus. Cytokinesis is the dividing of the cytoplasm. I'm going to go into a little bit about life cycle. So a life cycle is the reproductive history of an organism from conception to the production of its own offspring. Thank goodness I cut out plant reproduction every year. I do not like talking about plant reproduction. Plants just kind of bore me in general, and I know some biology teachers would probably like get so mad that I said that, but I'm just more interested in like animals and humans and stuff like that. So I've cut that out, but that's kind of why we would talk about this, is that we would talk about reproduction in plants, but we're just going to talk about it in humans. Fertilization and meiosis alternate. All right, in the sexual life cycles. Meiosis is the cell division that reduces the number of chromosomes from diploid to haploid. So what I need you to know here is obviously this definition, but I need you to know, what is this 2N and what is this N right here? The 2N becomes N. 2N means diploid, both sets of chromosomes from both parents. N means only one set of chromosomes. So 2N is diploid, N is haploid, all right? And gametes are haploid. Fertilization, I just need you to know the definition. When sperm and egg fuse to form a diploid cell, is fertilization. That diploid cell is called a zygote. A fertilized egg is called a zygote. A zygote then divides by mitosis to make a multicellular diploid organism. So I hope the process is kind of coming together for you. Meiosis makes sperm and egg. Sperm and egg fuse together through the process of fertilization. It forms a zygote. The zygote then does mitosis to form a multicellular organism. Within that multicellular organism, meiosis occurs and sperm and egg are made. I have a real good picture here for you. Here's a woman, here's a man. She has ovaries, he has testes. Inside of her ovaries, meiosis is occurring to form eggs. Inside of his testes, meiosis is occurring to form sperm. So you got your woman with her eggs, your man with his sperm. Here is the haploid egg, here is the haploid sperm. Boom, they come together, fuse, they form a diploid zygote. Both sets of chromosomes, diploid. Through the process of mitosis and development, it forms a big human. It's so beautiful, right? And then the process just goes over and over again. All right, so hopefully you kind of understood that a little bit. What makes meiosis so amazing and so great for humans is because it creates genetic variation. Because of that crossing over, you have sperm and eggs that are all unique, not identical, okay? Genetic variation in organisms is a really good thing. And so if you get anything from this lecture, this will be the most important thing that I say. Hopefully you're still listening. Genetic variation is a platform for evolution to occur. I hope I worded that correctly. Platform. I don't know if that's the right word. Cannot have evolution if you don't have genetic variation. That's what I meant. All right, have to have genetic variation in order for evolution to occur. When we get to evolution, I will bring this up again and I will show you why that is. Because, well, I'll just tell you right now. If you have all clones and they're in an environment that is not conducive to their survival, they're all going to either die or they're all going to either live. If you have genetic variation among those individuals, then the ones that are most fit for the environment survive, reproduce, pass their genes on, and pretty soon the gene pool changes. If you didn't catch what I just said, that's okay. I just need you to know that genetic variation is the basis for evolution. So how do we get genetic variation? In animals, in humans, crossing over, talked about that a lot. And then you have this thing called independent assortment of chromosomes. Students kind of get confused on this. Independent assortment of chromosomes. Remember I said homologous chromosomes pair up. In this picture you have both from the father on the left side and both of all the chromosomes from the mother on the right side. It's just as likely it could be this. It doesn't matter. Like, it's just random chance that the chromosomes from the father end up on one side and the chromosomes from the mother end up on the other side. And you can see as the pictures proceed, it makes a difference. If they line up like this, with the paternal chromosomes here, maternal chromosomes there, through the series of divisions, you get that outcome. Well, let's say just on this bottom set right here, they switched places. And there's a 50-50 chance, right, that would happen. Do you see how the outcome is completely different? So independent assortment of chromosomes is the random chance that homologous chromosomes line up the way that they do on the metaphase plate, with paternal or maternal being on the left or the right side. Hopefully that made sense. It's a random orientation of homologous pairs in metaphase 1. And then the third way that you get genetic variation in humans is random fertilization. Any sperm, any egg, there's a 1 in 64 trillion chance that you came to be. Isn't that so amazing? You're just so unique and special. All right. I don't think you needed to write anything down for that one, but just so you know. Random fertilization, any sperm, any egg. So if I asked you, what are the 3 sources of genetic variation in humans? You would say crossing over, and you would know what that meant. Independent assortment of chromosomes, and you would know what that meant. Random fertilization, and you would know what that meant. All right. The last thing I want to do before I end this, because I'm going to save karyotypes and those other disorders, chromosomal disorders, for next time. I just need you to understand the differences between mitosis and meiosis. All right. The picture shown here, mitosis is on the left. It's the division of somatic cells to make clones. This is what your skin does when you get cut or something like that. Over here on the right side is meiosis. This is the very specific process that occurs in the testes and the ovaries to make sperm and egg. So I need you to be familiar with these pictures. If you were to be shown one of these and I said, is this mitosis or meiosis? You need to be able to tell me. So let's jot down a few differences just to summarize everything that we've talked about. Both involve the division of a cell and the division of a nucleus. That's what they have in common. Mitosis is going to occur in somatic cells. Meiosis occurs in gametes. Mitosis involves 1 division. Meiosis involves 2. Mitosis results in 2 diploid daughter cells that are identical. Meiosis results in 4 haploid daughter cells. Clones identical, genetically different. Less than 1 in 8 million are alike. Mitosis occurs from zygote to death. So when a sperm and egg fuse and form a zygote, mitosis is going to occur until, if we're talking about humans, the human is deceased. Meiosis, this is kind of an interesting. Females before birth, follicles are formed. So basically meiosis has occurred in females before birth, which is pretty interesting. And then mature eggs are released when she hits puberty. And the purpose of mitosis is for growth and repair, and the purpose for meiosis is reproduction. You're like, wait a second, why didn't you talk about meiosis in males? That's because it's just like happening all the time, like forever, apparently. So, yeah. So in females, it happens before birth. All of her eggs are made. Males, they're just gonna make sperm forever, I guess. I mean, of course, with old age, it starts to slow down, and the sperm start to be, not quite like right, which is why they even say after the age of 35, a male has a higher risk of having a child with chromosomal abnormalities. And that's such a great place to stop, because tomorrow I'm going to talk about chromosomal abnormalities and some really interesting disorders. Okay, leave your questions at the end of the lecture, and I will see you guys later.